Hello friends, welcome to the channel. Hope you all are good. The topic of the day is vaccines and immunization need in present scenario. So nowadays we are going through the pandemic of COVID-19 and every day we are coming across the terms like immunization and vaccination. So it becomes important to understand the meaning of these terms and how or why the immunization and vaccines are so important in the present time. So firstly, let us understand these terms. Immunization, this is a process in which an individual's immune system is fortified or boosted up to fight a particular disease or an infectious agent. Similarly, vaccination means the act of administering a vaccine to an individual to boost up its immune response to a particular disease or an infectious agent. Therefore, relating both these terms, immunization and vaccination, we can say that the act of vaccination induces immunization in a person. Now, what are vaccines? We are going to understand in this lecture, but we should note that the vaccines save millions of lives every year. That means millions of people survive just because they are vaccinated. So, what are these vaccines and how they work? We are going to understand and study in this lecture. Now, first of all, let us understand what a vaccine actually is. A vaccine is a biological preparation that provides active acquired immunity to a particular disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe, its toxins, or one of its surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and keep a record of it so that the immune system can more easily recognize and destroy any of these microorganisms that it later encounters. A vaccine may contain live weakened microorganisms, or it can be made from dead microbes. On the basis of its types, vaccines can be of several kinds, like first are the killed vaccines. These vaccines contain killed organisms that were previously virulent. Examples of killed vaccines are influenza, cholera, bubonic plague, polio, hepatitis A, and rabies. The killed inactivated vaccines are produced by growing the pathogen under suitable conditions. The pathogen is further purified and treated with heat or chemicals so that it is inactivated, but it is still immunogenic. Next are the attenuated or the live vaccines. These vaccines contain live attenuated microorganisms. Attenuated means weakened microorganisms. The examples of these vaccines are live Mycobacterium tuberculosis vaccine, live attenuated vaccine containing strain Yersinia pestis EV used for plague immunization. For the production of live attenuated vaccines, the wild virus replication is done in unfavorable conditions. The process is repeated several times to produce an attenuated or weakened strain that is unable to cause a disease. Next comes the toxoids. The toxoids are inactivated toxic compounds in cases where these, rather than the microorganism itself, cause illness. Examples of toxoid based vaccines include tetanus and diphtheria. Next comes the recombinant vaccines. The recombinant vaccines contain either a protein or a gene encoding a protein of pathogen origin that is immunogenic and critical to the pathogen function. Genes isolated from a pathogen encoding major antigens can be inserted into non-virulent microbes. Such recombinant microbes serve as vectors replicating within the host and expressing gene product of the pathogen. Now let me introduce you with the production of anti-hepatitis B vaccine. The hepatitis B virus belongs to the family Hepatnaviridae. It contains a circular double-stranded DNA. The vaccine against this virus is prepared by cloning the HBsAg, that is the hepatitis B surface antigen gene of this virus in the yeast vector PMA56. These genes are cloned in the yeast vector with the help of enzyme EcoR1, that is E. coli restriction endonuclease 1. The hepatitis B virus surface antigen gene is joined to the ADH promoter cloned in the yeast vector. Now, this recombinant plasmid is further inserted into the yeast cells. These cells are multiplied on the tryptophan free medium and the transformed cells are selected. Further, these transformed cells are cultured. Further, the cloned yeast cells are selected for the expression of HBsAg gene. These cells are now sedimented. These are further lysed and purified to separate the HBsAg particles. The inserted gene sequence expresses and produces particles similar to 22 micrometer particles of the hepatitis B virus, as these particles are produced in the serum of hepatitis B virus patients. The expressed HBsAg particles have similarities in structure and immunogenicity with those isolated from HBV infected cells of patients. Its high immunogenicity has made it possible to market the recombinant vaccine against the hepatitis B virus infection. So this is how the anti-hepatitis B virus vaccine is produced. That is all for the day today. Thank you.